My name is Presley. Like Leah said, I'm in Warren Chan's lab, and this is one of my projects, liposome imaging in optically clear tissues. Okay. So nanoparticle delivery efficiency to tumors is very low. What I mean by that is that in 2016, a meta-analysis was conducted of the cancer nanomedicine field. And what it found was that even with 10 years of research dedicated to improving the delivery of nanoparticles to tumors, the median delivery efficiency did not improve past 0.7% of the injected nanoparticle dose, meaning that only 0.7% of injected nanoparticles were actually accumulating inside of the tumor. In a follow-up study, they broke this down even further and showed that only a very small fraction of the initial nanoparticles that are injected actually reach cancer cells, which is often the target of therapeutic agents. In terms of practical numbers, these percentages really relate to the total number of nanoparticles that are accumulating in cells. So if we inject 1 million nanoparticles to begin with into a mouse, we're only really expecting 7,000 of those 1 million particles to accumulate inside of the tumor, and only 14 to actually reach cancer cells and accumulate there. And what this suggests is that in order to design better therapeutics, it's really important for us to first understand what factors are influencing this nanoparticle distribution and uptake inside of a tumor. And one of the methods that we can use to begin to study this is a combination of three-dimensional microscopy and tissue clearing. A combination of these methods is really great because it allows us to obtain large volume images of a tumor, which we can then use to study distribution of nanoparticles with respect to a lot of the biological structures present inside of a tissue. So for example, we can look at how nanoparticles are distributed with respect to features such as blood vessels inside of the tissue. Unfortunately, these techniques are not currently compatible with all nanoparticles or other administered substances. And in particular, the tissue clearing process is not compatible with liposomal nanoparticles. And so if you take, for example, this clearing procedure, which is Clarity, developed in 2013, Clarity has a three-step process. We start by infusing the tissue with a solution containing formaldehyde and acrylamide. And what's really important here is that these chemicals react with the primary amine groups present on proteins to form a protein complex. These protein complexes then get polymerized such that the proteins in the tissue get cross-linked together, and we're able to actually form a tissue hydrogel hybrid where essentially all the proteins are cross-linked together, and this locks into place the biological structure of the tissue. Then, in order to render the tissue optically transparent, which is needed for our large 3D images, the tissues are treated with strong detergents such as SDS, which then transport through the tissue and actually remove lipids, which renders the tissue transparent, but also degrades our liposomal nanoparticles, and is the main reason why this process is not compatible with liposomes. So in order to get around this, we developed our project concept, which really asked the question, what if we could attach some sort of marker to the surface of a nanoparticle, which was capable of being cross-linked directly into the tissue the same way that proteins are? Then in theory, even if the particle itself gets removed, this surface marker should be retained because it's cross-linked into the tissue, very much like the proteins. And so this is the concept we worked with, and it led us to design a nanoparticle tag, which is termed REMNANT for retained molecule and nanoparticle tag. And REMNANT is a really simple design. Essentially, it's a surface marker that we can attach to a nanoparticle containing multiple primary amine groups. And if you'll recall, the primary amine group is of key importance here because it can be reacted with formaldehyde and acrylamide to be cross-linked into the tissue the same way as proteins. It's also really handy in the sense that we can easily conjugate dyes to these amines using NHS chemistry, which in theory should allow us to have both a fluorescently labeled tag that's cross-linked into the tissue. And so with this design criteria in mind, there were two main factors we needed to account for to ensure that this tag was viable as a nanoparticle labeling strategy. First and foremost, it should not alter the distribution of our nanoparticles inside of the tissue, and it also has to work to allow us to visualize liposome distribution. To begin testing the tag, we wanted to ensure, like I said, that it doesn't impact nanoparticle distribution. And we confirmed this by looking at the formulation of two different nanoparticle types, essentially a liposome without our tag, so with only a PEG present on its surface, and we compared that distribution to liposomes with our remnant surface tag. Inside of histology images, we looked at the distribution of these particles away from blood vessels and measured the distance that these liposomes traveled. We noted that there was no significant difference in the distance traveled from the vessels between our remnant liposomes and our control pegylated liposomes, confirming for us that the remnant was not impacting the distribution of these particles. We then went on to test these two designs inside of cleared tissues. And in the first case here, we have liposomes where they have just a lipophilic dye present inside of them. However, they don't have remnant. And we compared that to liposomes with our remnant surface tag present. And as expected, you can see that in the case where there's no remnant present, the liposome gets fully degraded during the clearing process. And as a result, we're unable to begin to study its distribution inside of the tissue. However, with remnant present, we have signal retained and we can begin to map the distribution of our nanoparticles with respect to these biological features, such as the blood vessels shown in this image. Now that we were successful in confirming that we could use remnant to map the distribution of nanoparticles in tissue, we wanted to take this project one step further and see if we could begin to use remnant to study features of our liposomes. We started by looking to see if we could measure liposome degradation rate inside of a tissue. The way we did this was by encapsulating a fluorescent labeled small molecule inside of our liposomes and comparing the signal of that small molecule to the signal of our surface remnant tag. What we were able to show is that over short periods of time, so between a half hour and 24 hours post-injection of our nanoparticles into a mouse, we see a significant degradation in the fluorescent signal of our nanoparticles. And so this is measured by looking at the fluorescent signal of our remnant and comparing it to the cargo inside of the particle. So we see a steep decrease in the correlation coefficient between these two signals, suggesting to us that the particles were being degraded. Now that we have a method of studying the degradation rate of the liposomes, we wanted to look one more step further and see if we could compare the degradation rates of our liposomes when we altered their composition. So we did this by testing 3 different nanoparticle designs, essentially by varying the saturation of our lipids and the relative percentage of cholesterol present. We saw that in the case of highly saturated lipids with high amounts of cholesterol, we had very stable liposomes at 1 hour post-injection. However, in the case of either low cholesterol or unsaturated lipids, our liposomes were substantially less stable, suggesting that they are degrading much faster and would have a faster cargo release rate. To study the impact of these release rates on the therapeutic efficacy of these particles, we encapsulated inside of these liposomes a small molecule, chlorgenate, which is capable of killing tumor-associated macrophages. What we did was essentially inject these particles with the 3 different formulations inside of a mouse and study the percentage of the macrophages that died as a result of being treated with these different release rate formulations of liposomes. We were able to show that in the case of our highly saturated and more stable liposomes, we saw substantially less macrophage death, as shown by the green signal here in this image. However, when compared to our faster release rate or less stable liposomes, we see substantially more macrophage death, suggesting that understanding the release rate of these particles is necessary and an important factor for designing better therapeutics. So to summarize, we designed REMNANT, which now makes it possible to study liposome distribution in cleared tissues. And it also allowed us to determine the impact of both liposome composition on degradation rate and their corresponding therapeutic efficacy. And so these are really the first steps that are needed towards designing more effective therapeutics for cancer nanomedicines. And with that, thank you all for listening, and I will take any questions.
Thank you, Presley. Presley, could you go back to slide 10 and tell us about that, more details about that experiment?
Yeah, so in this experiment, what we're essentially doing is encapsulating a fluorescently labeled small molecule inside of our liposomes. And to the surface of our liposomes, we're conjugating our REMNANT tag. In the case of these two different molecules, they're both labeled with independent fluorophores, and we're measuring the correlation coefficient between these two fluorescent signals over time. So what we see is at short time points, we have a lot of this cyan color, which is essentially representing a high correlation between our two fluorophores, the internal and external signals. However, at higher time points, we see that these signals become very unique in the sense that we have very distinct blue and very distinct green signals, but much less of this cyan color, suggesting that our liposomes have been degraded and that the cargo has been released.
Great, thank you.